they are coming later on lah Okay, never mind. Okay, I need to make sure every one of you join us today, right? So the last part ni quite complicated. Actually, you need to remember many things lah, many things, what is happening, in terms of how on this topic energy metabolism, how the food nutrient that we consume tu kan from the chemical energy being converted, being converted into ATP, the cellular energy. Alright, let me share screen. For those friends yang tak masuk lagi, boleh tak you panggil to join us as soon as possible, right? Okay, nampak Class, can you see my slide? Okay.
Nampak
okay, I make it slideshow. Right. Okay, nampak Moving. My slides moving
Moving, Doctor.
Okay. Our outlines, okay, we want to continue. So we have touched on the introduction, a little bit overview of metabolism, and the overview how food energy is transferred to cellular energy. So this is where we are going to look at the overview, the overall metabolism of the process, the biochemical pathways, and the cycles that involve, starting from the food, after being digested into the small molecules. So now we want to look at how the small nutrients, small molecules, the glucose, the fat and glycerol being splitted right from the digestion system from the previous topic that we have touched, discussed, and as well as the proteins. But since carbohydrate is the first, the major energy source, and followed by the fats. So this is where we are going to look at how the chemical energy being converted through all this biochemical process. And for beta oxidation, we are going to look at the cycle of beta oxidation, the breakdown of the fats, right? And then this is where we want to look at how the energy can be extracted from the carbohydrates, so-called glucose oxidation, or other terms so-called, what class? The glucose oxidation, the breakdown of the glucose, or scientifically termed as what class? Glycolysis. Ni nak exam dah ni, Glycolysis, okay? So it's another word for glucose oxidation or breakdown of the glucose, and then an extraction of energy from lipid. So in this case, fat, I want to, because the fat being splitted from its backbone of the glycerol, remember, in our intestine, right? Split in the form of micelles, and then being transported into the blood system until reaching the cell, the cellular, the body cells. And this is where the fat, because the glycerol, the 3-carbon glycerol can be converted directly into pyruvate and 3-carbon, because it's a small molecule. But the fats, so-called fatty acid, being further oxidized through the oxidation of fatty acid, also called beta oxidation. And why beta? So we are looking at that. Okay, so we have done this, the overview of the metabolism, consisting of both catabolism, or the breakdown or degradation, or anabolism, which is the biosynthesis. And then the metabolic pathways, when we talk about the metabolic pathways, it's always being involved with the presence of the metabolites. This can be as a substrate, or any products, considered as the metabolites. All right. So we have done this, both catabolism and anabolism. And then we have done this, we have touched what are the chemical energy being transported into ATP. So this is both catabolism, which producing this, the high energy, the high chemical energy, which are being utilized to the anabolism, to provide or to build up the cell macromolecules, right? Other than being converted into energy, so they are also being utilized for biosynthesis, biosynthesize back to the macromolecules to build up our body structure. All right. So we have done this. I have mentioned before this in this topic. So in this topic, I want to move. Okay, not right. Okay. In this topic, right, we want to look at there are 3 stages. Actually, when we want to look at the how the energy from the chemical energy, from the food nutrient that we consume, being converted into cellular energy. So involve 3 stages. I have mentioned before from the previous class. Where the stage 1 involve the digestion, absorption, and transportation. So we have done this from the previous topic. Okay, we have done this from the previous topic. Very important one. So this energy metabolism, where is the stage? We continue on the stage 2. So now we are actually focusing on the stage 2. Okay, where is the continuous from the transportation, meaning that the small molecules being digested in the small intestine and dekat lumen site though, remember lumen site, we are all the small molecules are being digested through digestion absorption. Okay, transported, okay, will be transported or travel through across the small intestine through the bloodstreams and reaching the cells, the body cells. Okay, this is where I want to look at the stage number 2, where the small molecules will be further broken down to a few key metabolites. The main metabolites is the acetyl-CoA, acetyl-CoA, which is the so-called very significant, very significant molecules or intermediate metabolites, metabolite perantara, intermediate metabolite, metabolites, metabolite perantara to convey all the carbons into the Krebs cycles, to be further broken down in the Krebs cycle. Okay, this is very important. The breakdown of small molecules to a few key metabolites. So we want to look at how fatty acid and glucose being broken down. Okay, and followed by the stage 3, where all the electrons being produced from the glycolysis, from the beta oxidation, being provided or being further transported or further broken down through the Krebs cycle. Krebs cycle is the metabolic central pathway cycle, metabolic cycle, so-called Krebs cycle. Okay, to produce more electrons, to produce more electrons from fatty acid, amino acid, as well as the carbohydrate. Then only being combined to be converted into ATP through the oxidative phosphorylation. Okay, this is the final stage, right? At the different location sites. Okay. This is a better overview. Okay, how the chemical energy being transported into ATP. So basically, you can use this very important flow chart for you to remember what are happening, what are the biochemical pathways or cycles available or occurs in this process. So we have done on the stage 1, digestion, absorption, and transportation. So this is where is the linkage between the stage 1 and the stage 2. Right, so the stage 2, now we have fatty acids. Fatty acids are normal lipids because of being broken down. Fatty acids, glucose, and amino acids. Okay, the main target for the breakdowns through TCA cycle. Right, so the stage number 2, we want to look at today. Continue. The breakdown of many small molecules of fatty acid, glucose, and amino acid into the Krebs cycle or citric acid cycle. Okay, this is where the acetyl-CoA must be produced first from the breakdown of fatty acids through glycolysis. Through the beta oxidation, and the glucose will further glycolysis and amino acid, of course, through the deamination, transamination, there are many other metabolic pathways to convert into acetyl-CoA. Yeah, 3 carbon to acetyl-CoA. How many? 2 molecules of pyruvate being converted into 2 carbon of acetyl-CoA. Acetyl-CoA has 2 carbon. Acetyl-CoA with the coenzyme A. All right. Okay, this is where the acetyl-CoA is a very important metabolite which convey all the electrons from the glycolysis, from the beta oxidation as well as deamination into the TCA cycles, being through 10, we call it 10 8 enzymatic reaction. So in the TCA cycle or Krebs cycle, the acetyl-CoA will be conjugated, combined with the substrate here, oxaloacetate, to further next 8 enzymatic reactions to produce all the electron carriers. And this is what I mentioned previously, NADH, NADH, FADH2, okay, and then we have GTP here, okay, as well as 2 molecules of CO2. Okay, so all these electron carrier carried out or transform into so-called the electron carrier, so-called NADH, right? So NADH, FADH2, NADH, nicotine adenine. You have I have mentioned before this. You need to remember nicotine amide, okay, adenine, dinucleotide, and FADH, flavin, adenine, dinucleotides. The other electron carrier, like a shuttle, energy shuttle, okay, that carries all the electron from the Krebs cycles, okay, and from the previous glycolysis, beta oxidation, and deamination into the other location from the mitochondrial matrix, which is occurring across the Krebs cycle, into the inside or inner mitochondrial membrane. We are looking for the later on. So this is where all the electron being transported through the electron transport chain. Okay, so all the proteins embed in the transmembrane. Okay, and they have involve 4 very important embed protein complex. Complex 1, 2, 3, and 4. Until it reaching the process of so-called chemiosmosis to convert. So we're looking at the stage 3 also, complete transfer, the final stage of the completing transformation of energy to ATP. So I will show you a few times, the repeated time, to show this kind of overview. Okay, just the same. So you need to remember the process that involve the biochemical process which involve at the different location site. Right. So as I mentioned, the acetyl-CoA production after the small molecules being further broken down through glycolysis. For example, the glucose undergo glycolysis to produce the 3 carbon, 3 carbon of the pyruvate. Pyruvate or pyruvic acid, it has 3 carbon molecules. Fatty acid undergo beta oxidation to produce also the process into acetyl-CoA. So same goes to the amino acid, will be further processed through the deamination to produce the acetyl-CoA. So this is very important of the formation of the acetyl-CoA before being further broken down or entering the Krebs cycle. All right. Okay, so we're looking at one by one the glycolysis first line. So this is where the pyruvate is the intermediate metabolite, just I mentioned before, before converted into acetyl-CoA, right? So the pyruvate is very important to be produced first from the 1 carbon, for 1 molecule of 6 carbon, 1 molecule of 6 carbon glucose being split into 2 molecule of 3 carbon pyruvic acid. So 3 carbon pyruvate and another one is 3 carbon pyruvate. They will be split into 2 molecules, which further broken down. So this is where we want to look at later on. Okay, so where the acetyl-CoA being transformed into the, so the first location where the glycolysis occur is kat mana class? Where is the process of glycolysis? You have mentioned that glucose being transported kan from the through the bloodstream into until reaching the blood cell into the body cell. So now is in the glucose in the cytoplasm. Class, cytoplasm, right? This is where the glycolysis occur. Okay, until it produce the pyruvate. And then the pyruvate will be transported into the another location. Another location site. Where? Kat mana? To produce energy ni. Very important, very famous location site. What is the organelle? The mitochondrial matrix, not only mitochondria, because don't confuse. The mitochondrial matrix, this is where the all electron being produced before being transported into another sites of the matrix. Okay? The internal, internal or so-called internal membrane, mitochondrial matrix. The mitochondrial membrane. So different with the location sites of the mitochondrial matrix, just like the cytoplasm of the matrix cytoplasm of the mitochondria. So mitochondria is an organelle, organelle in the cells to produce more electrons. Okay, so the first location from the where the glycolysis occur in the cytoplasm of the cell. Don't make mistake lah. Very important. The first stage usually must be in the cytoplasm. And when they converted into acetyl-CoA, then acetyl-CoA can be transferred into mitochondria matrix, cross into the matrix of the mitochondria organelle. Okay, of course we know lah, the mitochondrial, the organelle of the mitochondria. Okay, so this is where the further oxidation being occurred, through 8 enzymatic reactions, right? Through the glycolysis which will involve 10 enzymatic reaction. Okay, then for the TCA or Krebs cycle or citric acid cycle involve 8 enzymatic reaction to produce all the electron carrier. This is very important for every one of you as a biochemist. You need to remember what are the products being produced from the one cycle of this Krebs cycle. The central metabolic pathway, very important. And this is where the electron carrier in the form of reduced electron carrier, reduced form, reduced. Okay, so become being oxidized, so being reduced. In the form of electron carrier in so-called NADH, nicotine amide, adenine dinucleotide, and flavin adenine dinucleotide. So this both NADH, FADH2, as well as the 2 molecule of CO2 and 1 GTP here, somewhere here, 1 guanosine triphosphate. Okay, being their electron carrier being the electrons being transported into the next location, so-called after mitochondrial matrix, where all the electrons being produced through the Krebs cycle, will be further transported into the mitochondrial membrane. There's no matrix, the Krebs cycle occur. And then for the electron transport chain, where the oxidation phosphorylation, oxidative phosphorylation involve the both electron transport chain as well as the chemiosmosis. Okay, nanti kita tengok, we are looking at later on. Okay, this is where all the electron being transported ataupun being final completing their transformation into ATP. Okay, there are 3 different locations. Okay, class, okay, this is only the overview. Alright, so you should be able to remember lah, because as a biochemist, we need to remember. Okay, how the energy being transported. Okay, this is another so-called flow chart lah, the flow chart. The process of glycolysis, okay, which produce the electron and protons. And release the 1 molecule of CO2 and water molecules after glycolysis. right? And then when it will convert into acetyl-CoA, so acetyl-CoA will further broken down into this process so-called Krebs cycle or citric acid cycle to produce more electrons, to produce more electrons in the form of this electron carrier or energy shuttle, so-called NADH and FADH2. This is for the process of breakdown. Okay, so NAD or FAD is in the form, or NADP is the form of the so-called biosynthesis pathway lah. Okay, so this is oxidized form. This is the reduced form. So NADH and FADH2, they are the electron carriers which carries all the electrons from the glycolysis and from the Krebs cycle, okay, into the next location of mitochondrial membrane, okay, which will be further or undergoes the electron transport chain as well as the chemiosmosis lah. This is only the flow chart lah, so that I'm keeping repeated, keeping repeat the flow chart, the overall process, so that you can remember, easily you can follow lah. Okay, basically this is the process. Okay, so by energy of ATP, so we know that the energy is trapped is in the form of ATP, adenosine triphosphate, the energy currency, and ATP is generated during glycolysis. I have mentioned previously, okay, generated. We'll see, one by one, what are the product and how many ATPs being utilized, in the glycolysis and being produced. Okay, and then only we will get the net of ATP, the numbers of net ATP, to ATP to can. And then also oxidative phosphorylation, okay, which involve both the electron transport chain, and as well as the chemiosmosis, okay, through the electron carrier, okay, nicotine, amide, adenine, dinucleotide, sorry, class, dinucleotide, this is an NADPH, so nicotine, amide, adenine, dinucleotide, and ADH is dinucleotide. Or flavin, adenine, dinucleotide. Okay, both NADH and FADH2 are the electron carrier or energy shuttle. Alright. Okay, so these are generated during reaction of both glycolysis and TCA cycle. In the form of free energy, ATP is required to perform various ATP-dependent reactions. Of course, many biochemical reactions, even though we have done previously in food biochemistry, since early in the morning when we wake up early in the morning, or in anything, any work you want to do, we need to have a little bit energy, depending on what kind of movement you want to do, any types of work you want to do, we'll start to move our body muscles, so we need the presence of enough ATP, right, and many other anabolic mechanism. Okay, so this is only the molecular structure of the ATP, adenosine triphosphate. Okay, it's actually in the form of what class? What kind of biomolecule is this? ATP is actually a molecule of what? Nucleic class?
Nucleic acid.
Nucleic acid, and its monomer is, so the ATP is the monomer, a monomer of nucleic acid, so called nucleotide. Okay, nucleotide. We have touched before this, the structure of ATP, adenosine triphosphate. Okay, we have seen this is a ribose. There are 3 elements in one monomer of nucleotide. We have ribose at C1. We have this is a so-called the nitrogen base, nitrogen base, right? At the carbon number 1, and where is the phosphate groups? At the carbon number 5. This is where the carbon number 5 in the phosphate group will be continuously linked to the next nucleotide, next monomers of nucleotide until become the long sequence of nucleotide, so-called nucleic acid lah. So this is one monomer of nucleotides. So the phosphate group will have the linkage with the next monomer of nucleotide through the phosphodiester bond. Phosphodiester bond. Okay, so this is only one monomer of nucleotide, so-called this ATP. Okay, so can be broken down into inorganic phosphate, one molecule of phosphate with the ADP, adenosine triphosphate. Okay, so this is process of the hydrolysis lah, the breakdown. So the ATP also can be broken down, right? So this is where we have touched before, we have learned during our food biochemistry in one topic, the muscle contraction, right? The muscle contraction where the ATP is needed, right? Remember that? To produce or to provide the energy for the head, the head of the myosin. If you still remember lah, remember tak myosin? This is apa? Myosin. The myosin, the thick one. Remember tak? And there's one more. The myosin, the head, the head of the myosin tu kan? Remember? The muscle. Tak ingat kan, the muscle contraction, right? So the muscle must be activated first, in the presence of the ATP, and it will start to contract or touch to make the linkage. Remember yang tu? Remember okay. So everything or any movement you want to do, our body want to start with, must be the presence of ATP. Okay. Okay, this is where the overview of the process, how the glycolysis produce their electrons, and further undergoes the Krebs cycle to produce more electrons. So I mentioned electron and proton together. The proton is H plus. Here are the protons. It's very important. We want the important part of the protons. We will see later on. Through this electron transport later on. Okay, so the glycolysis will convert into 2-pyruvic acid. I have mentioned before this. One molecule of 6 carbon of glucose will be split into 2 molecule of pyruvic acid. Okay, 2 molecule of 3 carbon pyruvic acid. So the pyruvic acid or pyruvate, it has 3 carbon. Okay, 3 carbon. So 3 carbon and 3 carbon. All right, being split from one molecule of 6 carbon of glucose. Okay, so from 1 molecule of 6 carbon glucose being split into 2, this is 2 molecules of 3 carbon of pyruvic acid. Okay, and then further transported into the Krebs cycle. Okay, so why to acetyl-CoA class? Because we have 2 molecule of 3 carbon pyruvic acid. So each molecule of pyruvic acid will transport, or we will convert into acetyl-CoA. And another one molecule of pyruvic acid will undergo the same process to form the acetyl-CoA. That is why we have 2 acetyl-CoA here. All right. So meaning that from 1 molecule of 6 carbon of glucose will further split into 2 molecule of pyruvic acid to produce 2 acetyl-CoA lah, because each pyruvic acid produce or transform into acetyl-CoA, 2 carbon acetyl-CoA, and another molecule of pyruvic acid also same, goes the same process to convert or transfer into acetyl-CoA. That is why 2 acetyl-CoA. So each acetyl-CoA will undergo into Krebs cycle. So the other one, acetyl-CoA also. So this is where the combination of two process two process. Since we produce 2 acetyl-CoA, so we'll undergo 2 Krebs cycle. So we want to look at how many total numbers of ATP being produced from 1 molecule of 6 carbon glucose. Considering these 2 molecule being converted, so become double lah. Okay, become double. right? Starting from the pyruvic acid. Okay, and further acetyl CoA. So, through the Krebs cycle, I have mentioned before this, we produce CO2. Okay, 2 molecule of CO2. So this is 4 cup CO2 lah because it's double, right? We have 2 acetyl CoA, right? And we further the same Krebs cycle, so it become double lah. Each Krebs cycle produces only 2 carbon CO2, 2 CO2 molecules. So in total is 4. Same goes to this, we have the Krebs cycle produce 3 molecule of NADH, and this is where it will double, so produce 6 NADH. One Krebs cycle produce 1 molecule of NADH, so it's double, become 2. Same goes to this guanosine 3-phosphate. Krebs cycle produce only 1 GTP, but it's double process, so producing 2 GTP. So all these electrons being carried or transformed in the form of NADH and FADH. Okay, so in total, before we converted through the electron transport chain, and finally being converted into the final product is the 38 of ATP. So 34 ATP from this glycolysis, and from the Krebs cycle. Okay, 34 ATP. So plus 2 ATP from GTP. So we will see the equivalent. The equivalent. 1 GTP is equal to 1 GTP is equal or equivalent to 1 ATP. If 2 GTP is equivalent to 2 ATP. And 2 ATP also being produced from the glycolysis as a net ATP. One glycolysis process produce 2 ATP. So we sum up 34 ATP plus 2 ATP and then plus another 2 ATP from glycolysis, and make it 38 of ATP in total. Okay, this is the overview, the whole process, how the glycolysis or how the glucose being oxidized through a Krebs cycle and the oxidative phosphorylation to completely produce 38 of ATP. Okay, class? Okay, let's look at one by one, the glycolysis. Okay, so this is quite keep repeating lah. I'm repeating the process so that you can easy to remember. Alright, so as I mentioned, glycolysis can be defined as the glucose splitting or glucose breakdown. This is an aerobic process occur in the cytosol. Okay, the student as biochemist, you should be able to remember the location sites in the cells. Focusing on the cytoplasm and the mitochondria. Okay, this is where the energy being produced. Right, so you must remember this location occurs in the cytosol or cytoplasm of the cell. It's the one metabolic pathways found in the living organism. Yes, the sequence reaction of that split the sequence. I mentioned before this. I keep repeating. From the 1 molecule of 6 carbon glucose. Okay, glucose has 6 carbon. As I mentioned, 1 molecule of 6 carbon glucose being split into 2 molecules of 3 carbon pyruvate. So the pyruvic acid has 3 carbon in their structure, but it has 2 because it being split from the 6 carbon, okay, of glucose. Okay, and the small amount of ATP. So the glycolysis consumes 2 ATP, meaning that the glycolysis, they have 2 stages. We'll look at later on. The first stage, also called the introduction phase. The first stage, we need the glycolysis need or consume 2 ATP. Okay. But produce in the second stage will produce 4 ATP, right? And the release of 2 molecules of NADH. That is why the net products of the ATP from the glycolysis is 2 ATP. Okay, the 4 ATP produced minus the ATP consumed. So the total net of ATP being produced by glycolysis is 2 ATP. You must remember, what are the products from the glycolysis? 2 ATP, 2 NADH, and 2 molecule of pyruvate, which being split from the 6 carbon glucose, and involve 10 enzymatic reaction, or 10 steps involve each reaction involve the enzymes. So 10 enzymatic reactions. Okay, class. 10 enzymatic reaction. But in the Krebs cycle, 8 enzymatic reaction. Okay, so you need to very important for students to remember what does it mean by glycolysis. What are the products? What are the products? The net products ni 2 ATP, the numbers to ATPs, how many enzyme involved? Okay, 10 enzymatic reaction. Don't worry lah. I want to ask regarding on the what enzymes, what is enzymes. They undergoes this. Don't worry, right? Okay, but you need to know the enzymatic reactions involved in the glycolysis. Okay, this is where you want to look at how one molecule of 6 carbon of glucose being split into 2 molecule of pyruvic acid. Okay, this is one molecule of pyruvate, 3 carbon pyruvic acid, and another one from the AP here, the hydroxyacetylene phosphate, also goes the same process to produce one more 3 molecule of pyruvic acid. Okay, let's look at this. This is the so-called the first phase, fasa pertama. The first phase from one molecule of 6 carbon of glucose, okay, need the presence of the ATP. So the first ATP I mentioned kan, the first phase of the glycolysis consume 2 ATP. And they require 2 ATP. So the first one, when the ATP is present, attached at the carbon number 6 to form the glucose 6-phosphate. And then isomerase enzyme, okay, isomerase, you know, right, the isomerase to isomer, to isomerize the glucose into or turn or switch into fructose. Isomerase actually turns the molecule structure without just maintain the numbers of the molecular structure. The molecular formula is just the same. The molecular formula is just the same. 6 carbon. Okay, from the glucose 6-phosphate attached the phosphate at the carbon number 6 and isomerize into fructose. The changes or the difference is only the position of the hydroxyl groups. The hydroxyl groups in this fructose 6-phosphate. Still the phosphate at the carbon number 6. But the position of the hydroxyl group. Okay, so the next one, the ATP, then the second ATP is being produced to attach at the carbon number 1. So the name is become fructose 1,6-biphosphate. Okay, this is the first phase of glycolysis, which consume 2 ATP, right? And then the fructose 1,6-biphosphate will further split. This is where the splitting of glucose being occur, where the fructose 1,6-biphosphate, it still has 6 carbon, being split into G3P. So this I always mention G3P, glyceraldehyde 3-phosphate, G3P, glyceraldehyde 3-phosphate, and another 3 carbon so-called DAP, dihydroxyacetone phosphate, DAP. So both G3P and DAP undergoes the step of 6, step number 6, number 7, number 8, 9, 10. Where class, okay, from this 3 molecule of G3P, glyceraldehyde phosphate, needs produce being broken down to produce 2 more ATP. It is step number 7 and step number 10. Okay, to produce 2 more ATP, to produce 1 molecule of NADH here. Okay, in the consumption ataupun the first phase consume 2 ATP. Alright, since at the second stage, both G3P and DAP produce 2 more ATP. From G3P produce 2 more ATP, while the other 3 carbon and the DAP also produce 2 ATPs. Okay, so this is the total number of production of ATP is 4. Okay, from the glycolysis, but the net ATP is only 2. Okay, so from 4 ATP being produced minus 2 ATP in the first stage. So the net ATP become 2 ATP. This is what I mentioned. The net product is 2 ATP. And 2 NADH lah. Because one ADH here and one more ADH under this process, under DAP process. So the same process as TP class to produce the pyruvate. Okay, so you got me class? You got me class? So far? How one molecule of 6 carbon being split into 3 carbon pyruvate acid. But itulah, it has two molecule of 3 carbon pyruvate acid here and here. Here is from the DAP lah. Okay, undergo the same process. Okay, so the total numbers of products of ATP is 2. Net ATP is 2 because from the 4, the 4 at the second stage producing ATP, right? From the second stage minus 2 ATP consumed at the first, from the first stage. So the net numbers of ATP is 2 ATP. Okay, boleh kelas So in the pyruvate, 3 carbon pyruvate acid, this is where the pyruvate acid will convert into acetyl-CoA before being transported into TCA cycle. Okay, class we just mentioned, we just go through first through the glycolysis. Okay, the very important step. Conversion the pyruvate acid from the 3 carbon pyruvate acid into 2 molecule of acetyl-CoA. Okay, before being further converted or broken down into TCA cycle. Okay, this is where the pyruvate acid, okay, in the presence of the oxygen, if we still have, for example, when we do a very extreme exercise, regular or quite extreme exercise or any activities, okay, where we need the more ATPs being produced. So depending if you are lacking the oxygen, we are suffering from the oxygen, okay, so it will undergo the lactic acid production by the lactate dehydrogenase. But in the presence of the oxygen, in production of, if we still have enough oxygen, so the pyruvate will be further broken down into TCA cycle. Okay, this is where you want to look at class from the pyruvate 3 carbon of pyruvate acid, okay, converted into two carbon of acetyl-CoA. Okay, why 3 carbon pyruvate acid become 2 acetyl-CoA? Because in the transformation process, it will release 1 molecule of CO2. So 1 carbon is released, and then leaving 2 carbon of the acetyl-CoA in the presence of the coenzyme A. Coenzyme A will convert or will conjugate, combine into the 2 molecule of acetic acid to form the acetyl-CoA. Then only can be transformed or can be transported into Krebs cycle. Okay, class So far you can follow me, class.
Yes, doctor.
Okay, this is where I mentioned tadi kan from the overview, this one. Okay, where each pyruvate kan from 1 molecule of 6 carbon glucose can produce 2 pyruvate acid. Betul tak, class? 2 pyruvate because it comes from this lah. Splitting from 1 molecule of 6 carbon into 3 pyruvate acid and another 3 pyruvate acids. So there are 2 pyruvates or 2 pyruvate acid produced from 1 molecule of 6 carbon being split. So 2 molecule of pyruvate acid. Okay, so each pyruvate will undergo or transfer into acetyl-CoA. So meaning that another one more pyruvate acid also undergo or transform into acetyl-CoA. So become 2 acetyl-CoA. Alright. So 2 acetyl-CoA, each acetyl-CoA will transform or transport into TCA cycle, meaning that there are 2 Krebs cycle involved. That makes a double calculation of ATP. Okay, class, you got me? If you want to ask, please ask. You can ask me. You can interrupt me whenever you want. Okay, all right. Still glycolysis. Okay, the expression that I have mentioned just now. Okay, where the first phase, this is glucose, the glycolysis process. Again, I repeat, right? So from one molecule of 6-carbon glucose, okay, consume need the 2 ATP as I mentioned tadi kan, from the first input ataupun from the first phase, fasa pertama okay, so 2 ATP is needed, right? And for the second phase, which is the energy payoff phase, release 4 ATP and 2 NADH. Okay, class 2 NADH, because it has been split into 3 GTP tadi kan, and DAP, which produced 2 ATP and 1 NADH. So it become double lah, become split. Alright, so both G3P and DAP undergoes the same. Okay, second stage ni. So the net ATP is become, okay, this is a net ATP, the net ATP. So the output for ATP molecules, 2 molecules of NADH, of course, the pyruvate become 2 lah, big split, the 3 carbon. So the net ATP is, okay, 2 molecule of pyruvate, and okay, 2 ATP because 4 ATP minus 2 ATP, okay, 2 ATP in the outputs, minus ATP in inputs. So the 2 ATP is the net, net number of products, as well as 2 NADH. So 2 pyruvic acid, 2 ATP and 2 NADH as electron carrier, they are the net products from the glycolysis. Okay, class, glycolysis. So the net ATP is only 2 ATP. Alright. Please keep in your mind, betul-betul. So easy to follow the next one. Okay, again, I show you one more time. Okay, the same, just the same, exactly the same overview just now from the first diagram tadi. Okay, the stage 1 and stage 2 tadi produce reduced electron lah. So NADH2, NADH and FADH2. Okay, NADH just now produced by glycolysis, 2 NADH, right? Electron carrier from the glycolysis. But in the Krebs cycle, it has 1 molecule of FADH2. So both NADH and FADH2 carries all the electron. Okay. Okay. So this is where the stage where all the NADH, FADH2 transform or transported into stage 3. Okay. Same. So you can the students should be remember lah the process. You can make it in the flow chart, or you can make it like this lah, diagram. Okay, this is where I mentioned where the acetyl-CoA being transformed from the pyruvate. Okay, from the glycolysis, produce the pyruvate 3 carbon. Okay, there are 2 molecules, right? 2 molecules of 3 carbon pyruvate acid, meaning that this is only one lah. In one molecule pyruvate which undergo or transfer into acetyl-CoA, 1 molecule of acetyl-CoA with 2 carbon, and undergo 1 cycle of the Krebs cycle. This is only one lah, but need to remember, okay, the pyruvate ni it has 2, so another 1 more molecule of pyruvate acid undergoes the same, transform into another acetyl-CoA. That is why it has 2 acetyl-CoA in the overview for the ATP, total number of ATP. And the acetyl-CoA, another acetyl-CoA also will further the same process of Krebs cycle. So that's why the numbers of ATP become double. So let's look at only one Krebs cycle. So it's okay when you want to calculate, just make it double. All right. So let's look at this, the pyruvate 3 carbon pyruvate. This is where I mentioned how come the pyruvate from the 3 carbon transform into 2 carbon of acetyl-CoA, because the process of this the so-called decarboxylation. The 1 molecule of carbon from the pyruvate being released into CO2. And at the same time it releases NADH, electron carrier together with the protons. to form the acetyl-CoA. Okay, then only the acetyl-CoA will enter the Krebs cycle. What is the location, class? Mitochondrial matrix. Or cytoplasm of the mitochondria. Usually we mention about as a mitochondrial matrix. Okay, so the acetyl-CoA now, to enter the Krebs cycle, it needs to conjugate or combine with the first, first substrate here, the oxaloacetate. Okay, the oxaloacetate is actually, this is the Krebs cycle is the cycle, right? The cycle, so it keep repeating. So the acetyl-CoA, 2-carbon acetyl-CoA will conjugate or combine with the 4-carbon oxaloacetate. This is kind of one of the metabolite from the Krebs cycle. Okay, as I mentioned, either substrate or so-called metabolites, intermediate ataupun perantara. So oxaloacetate 4-carbon will conjugate with the 2-carbon acetate to form the 6-carbon of citrate. As a biochemist, you need to remember it's very easy. So you can make your own mnemonic for TCA substrates. Citrate is the Krebs starting substrate for mitochondrial oxidation. So all these 8 citrate, the first substrate. Remember, that's why I put it mnemonic. Mnemonic. Citrate is the Krebs starting substrate. So the citrate is the first substrate. So the citrate is carbon and it isomerized into isocitrate. So citrate is the isocitrate. And isomerized lah. So there is the enzyme to isomerize it. And then the isocitrate will release the first one molecule of CO2 and one molecule of NADH. So this is electron carrier being produced, NADH, and together with proton. And the next one is starting substrate for mitochondrial, okay? Isocitrates, okay, starting substrates, isocitrate and alpha ketoglutarate. From the isocitrate K is the ketoglutarate. Alright, so release 1 carbon SiO2 to form the 5 carbon lah. That's 1 carbon. And from the 5 carbon here, release 1 more CO2. From the Krebs cycle, only 2 molecules of CO2 produced. Okay, but remember it has another Krebs cycle. Okay, become 4 lah. In order to calculate lah for overall ATP. But in the one Krebs cycle, okay, 2 molecules of CO2 only being produced. Okay, and then 1 more and NADH being produced. It's become starting here is a succinyl-CoA. Okay, become 4 carbon. And this is where I mentioned 1 GTP is produced. 1 GTP is guanosine triphosphate. Guanosine triphosphate. Another one nucleotide also. One nucleotide usually is used for the signaling molecule. Signaling molecule, right? But it is calculated. It is calculated into the overall ATP production. Okay, now succinate. The substrate here is succinate. Okay, for carbon. And here, this is where the first FADH is produced. Okay, the first and the only one lah from the Krebs cycle. Okay, so fumarate. The 4 here is the fumarate. Easier to remember. Citrate is the Krebs starting substrate. At least you remember what is the first from the conjugation between the acetyl-CoA and oxaloacetate. So the first substrate is the citrate. Right, so isomerized into isocitrate is easier to remember. And then from the fumarate, which after being produced the FADH as the second energy shuttle, energy carrier, will further undergo the mitochondrial oxidation. And after M is back to oxaloacetate by releasing 1 more NADH. So 1 Krebs cycle producing how many NADH class? 1 Krebs cycle. 1 from 3 carbon pyruvate, from 1 molecule, 3 carbon pyruvate undergo 1 cycle of Krebs. So 1 Krebs cycle producing, what are the products? What are the products?
Carbon dioxide,
How many molecules CO2? From 1 Krebs cycle produce 2 molecules of CO2, carbon dioxide. What else? NADH, how many?
One.
Three molecule of NADH. Okay. What else about the FADH? Produce one molecule FADH and one more GTP. Okay, GTP. So need to remember from products from the glycolysis and also products from the Krebs cycle in order for us to calculate, calculate the total activity. Okay, alright. Dah pukul berapa dah ni? Okay, kejap Hold on. Anak kejap minit. Okay, we continue. Okay, class, so far? Tak apa, you need to make your own revision lah later on. Okay, so yeah, we continue lah Right, so this is quite overall again from the glycolysis, deamination or beta oxidation, so producing the acetyl-CoA. So we have look at just now how the acetyl-CoA being transformed from the 3-carbon pyruvic acid. So 2-carbon acetyl-CoA will convey all their electrons from these pathways semua kat sini into the TCA cycle. Okay, 2-carbon acetyl-CoA just now it conjugate with the class apa? Acetyl-CoA conjugate with the carbon with the oxaloacetate with how many carbons? 4. Remember 4, and then producing 4 plus 2 producing 6 carbon of
citrate.
Okay, remember tu lah the next cycle. So there are 8 enzymatic reaction but the most important are the numbers. What are the products? What are the products produced from one cycle of Krebs or TCA cycle? Okay, so we have, I mentioned just now, okay, you have mentioned just now that there are 3 molecules of NADH and 1 FADH2 and 1 more actually GTP, release 2 molecules of CO2. So the reduced coenzyme, in the form of FADH and PH will carry the electrons and protons into the next stage. From the matrix. Okay, class, look at this. This is the organelle, mitochondria organelle. Okay, in the cells. So if you're looking at this, the mitochondria, it has the membranes. Okay, they has 2 inner membranes, the inner membrane, then outer membranes. So the inner membrane is the space, is the location where the stage 3 occur. The oxidative phosphorylation to complete the ATP production. This is where the process occur in a membrane. So the matrix is where the process of Krebs cycle the Krebs cycle occur to produce or oxidize the acetyl-CoA right into 3 NADH and 1 FADH2, right? So this reduced coenzyme will carry or transported into the inner membranes, inner membrane to apa tu kita panggil to transport electron. Dia nak pass to pass the electron that will that carried by this reducing enzymes. So the energy shuttle tadi kan, the energy like a bus shuttle occur, the drop all the electron there, drop the electron, and so the electron will be transformed into a series of embed proteins in the inner membrane. Okay, so remember the location site is very important. The flow tu so you can manage lah the way you study, the way you want to easy to remember. Okay, so okay, the final compound. So for the FADH3 and ADH from the oxidation into the Krebs cycle occur in mitochondrial matrix. So this is where we want to carry all the electron carrier FADH2 and ADH into the inner membrane site. Okay. Okay, again is another flowchart lah, sama je lebih kurang So now I mentioned FADH2 and ADH are reduced coenzyme lah. So when they release the electron, so the oxidized coenzyme kan, I mentioned tadi kan, this enzyme again carries all the electron. It's repeated lah. Repeated because this is FAD and AD. I mentioned before this, they are the oxidized coenzyme. But those carrier that carries electron and proton, so-called reduced one. Because the trimer, they will receive the proton and electron, so become reduced lah. Okay, so when they travel or transform into the inner membrane space of the mitochondrial membrane, the inner space of the mitochondrial membrane, mitochondrial inner membrane, okay? Mitochondrial membrane pun okay lah, membrane. So it will release all the electron, which further undergo so-called here, this is the process of oxidative phosphorylation class, oxidative phosphorylation, okay? So this is where all the protons and electron, okay, the proton will be pumped, they will undergo the pumping process of proton, okay, to produce or to make the gradient, the proton gradient concentration is different between in the mitochondrial in the membrane and the mitochondrial matrix. Later on kita tengok So and also the electron being transformed into the complex protein which embed in the membrane space. Okay, of course at the same time the production of the pumping of proton, okay, will involve the formation of H2O. Okay, they contribute H plus ni contribute to combine with the oxygen to produce water. So the water will be released in together with the ATP production. Okay, again lagi so I keep repeating so that student can remember lah easily. Okay, so again from the acetyl-CoA underscore the Krebs cycle class. Okay, so the products are here: 3 NADH, 1 FADH2, 1 GTP. Alright, and release 2 CO2, okay, 1 molecule water molecule and CoH lah, because coenzyme in the beginning kan, from the acetyl-CoA produce, okay, but the very important the product and NADH, FADH and GTP. Okay, there is in terms of calculation for overall ATP, so each one molecule of NADH, the electron carrier, this electron carrier NADH give or equivalent to 3 ATP. Okay, while for FADH2 equivalent with 2 ATP. So we need to calculate, we double, we times 2 or times 3 depending. Depending whether if an ADH, how many ADH we just times 3, and how many molecule of ADH we times 2, right? Okay, let's just look at this on top here. Now, from the Krebs cycle, produce products 3 NADH, 3 times 3 lah, because 1 molecule NADH, okay, give 3 ATP. So since there are 3 molecule of NADH, from Krebs cycle ni, we times 3 to form 9, to produce 9. And 1 FADH times 2, become 2 lah. 1 FADH equivalent to 2, become 2. 1 TTP, 1 TTP is equivalent with 1 ATP, so become 1 lah. So the equivalent is 12 ATP. 12 ATP per 1 cycle, 1 Krebs cycle, 12 ATP per 1 Krebs cycle. Okay, but remember, we have 2 Krebs cycle, kan? Because there are 2 acetyl-CoA, so it can become double lah, right? But what the most important, you need just to remember only 1 cycle, how many products, okay? How many products, and also from the glycolysis, how many products at the end. Okay, same goes to glycolysis also. From the beginning I mentioned tadi kan the calculation tu, tapi we will repeat where from the glycolysis also produce an ADH and ATP. So they will times this just like this same. But glycolysis doesn't produce FADH, only an ADH, right? To an ADH. Okay. Again, class. Okay, I repeat. I think this is the last overview. By explaining, this is where I want to show again, from the glycolysis, okay, from the Krebs cycle, and now I want to move to the next stage, the final stage, which involve the electron transfer and oxidative phosphorylation lah. Okay, this is where all the electrons will pass into the protein complex, complex number 1, 2, 3, 4, okay, until the pumping, which also involve the pumping of protons, okay, to make the gradient, of the mitochondrial matrix and inner membrane space become different, so it can pumping the proton back into the mitochondrial matrix. This is where it provide the energy, the pumping of proton provide the energy to release phosphate, organic phosphate. Kita akan tengok, we'll look at later on, okay, how the pumping of protons that make the different in the gradients, the gradients of the protons in between two spaces, mitochondrial matrix and in the membrane space, can give or provide the process of chemiosmosis, where the ATP synthesis or ATP synthase will release its ataupun the pumping down the protons into its rotor to provide inorganic phosphate lah. So it can combine with the ADP to produce ATP. Okay, at the same time, protons pumping contribute to the formation of water, as I mentioned before. All right. So what are what is the stage 3 though? The stage 3 or involve the oxidative phosphorylation, involve the series of enzymes which embed in the mitochondrial inner membrane. Okay. So in the stage 3, the process involve in between the mitochondrial matrix and the mitochondrial inner membrane saja. Okay. Mitochondrial inner membrane. Okay. Doesn't deal with the outer mitochondria, only the inner membrane which facing close to the matrix. Okay, where all the protein complex embed to transfer the electrons. So the protein complex number 1, 2, 3, 4, okay, involve this transformation of electron, transport, pass the electron. Okay, but for the complex number 1, 3 and 4 only, kita tengok 1, 3 and 4 only involve the pumping of proton, pumping of proton. Okay. Okay. So there is a ATP synthesis and other proteins which embed as an enzyme. For I mentioned complex electron transport chain just now, electron transfer through the electron transport chain. Okay, senang this one lah. Alright. The last one is ATP synthesis. Okay, class, if you're looking at this, this is the electron transport chain. The stage number 3, which involve, okay, all the NADH and FADH from the mitochondrial matrix, from the Krebs cycles, the products from the mitochondrial matrix, from the Krebs cycles being transformed okay, transported into the electron transport chain. So we can the electron transport chain here involve there are involved 4 protein complex of electron carrier. Complex number 1, complex number 2 here, complex number 3, complex number 4. So complex number 1 is the FMN. This is FMN. FMN is the flavin, according to the flavin mononucleotide. Flavin mononucleotide. But flavin mononucleotide, if you still remember, I think Dr. Badrin also covered this. It's a kind of vitamin B, vitamin B2, riboflavin. The word flavin is due to the flavin, riboflavin of vitamin B2. Okay, flavin mononucleotide. Okay, complex number 1. And CoQ, coenzyme Q, is a kind of cofactor. Coenzyme, coenzyme Q is an enzyme lah, protein. So complex 2, 3, and 4, they are the enzyme. Complex 3 are protein, the cytochrome B and C, and complex number 4, cytochrome A3. A3, like sometimes A3 or A3. So this complex number 1, the first complex of protein which embed. So all these proteins and riboflavin, complex number 1 is the vitamin B2. So this riboflavin which embeds in the embed in the membrane of the mitochondria, will receive the first electron from the NADH as well as the protons. Right? So NADH will first attach pass its electron in this complex number 1. So complex number 1 FMN receive the electron. Okay, together with the protons. So the protons will be pumped. This is where the function of proton here. So the proton provided by the energy shuttle of NADH from the mitochondrial matrix will pump into or cross to the next side, which is the intermembrane space. Okay, the stage number 3 involve mitochondrial matrix and the intermembrane space. Okay, the intermembrane space. Okay, so the inner, this one lah, the inner is the ETC tu, ETC embed dekat inner. So the ETC, the transport chain, it embed in the inner mitochondrial membrane. But the proton which pump from the mitochondrial matrix into across this inner mitochondrial membrane into the intermembrane space. Same goes to this. So the electron first, pass its electron from the NADH from the complex 1 to the complex number 2, coenzyme Q. Complex number 2, coenzyme Q. Okay. If you look at this, what is the different class from the complex 1, 2, 3, and 4? Complex Q, the complex 2 of the protein embed ni, doesn't involve with the pumping of protons. Just transfer, only collect the electron carrier from the complex number 1 and from the complex number 2 from FADH. So FADH pass its electron. So the CoQ, complex number 2, collect or receive the electron from complex number 1 and 2 and pass to the complex number 3, okay, cytochrome B and C, and further transported into complex number 4, right? So each complex 1, 2, 3, and 4, it has in details, it has its own function lah. There are a series of function of each complex differently depending dealing with their function towards towards the different types of molecules being transported in between this intermembrane space in the mitochondrial membrane lah. But here only you want to look at only the electron, the electron transport. Okay. So they have the other function lah. But in this case, the function of complex 1, FMN, CoQ from BC and AA3 is to transport or transform to pass the electron into the final complex 4 ni as the final electron acceptor. The complex number 4 is the final electron acceptor. Okay. Complex 1, 3 and 4 involve only 3 complex 1, 3 and 4 without or except for Q involve the pumping of protons. Involve pumping of protons, and it will contribute to the higher level, higher concentration of protons in the intermembrane space. This is make or contribute to the gradient, the concentration of gradient level of the proton pump in between intermembrane space and in the mitochondrial matrix. Okay, so the pumping of protons from complex 1, 3 and 4 increase the concentration of protons, which make the gradient, the gradient due to the different concentration from the different spaces. So the intermembrane space will have higher level of concentration of protons. What happens when the gradient level at the intermembrane space is higher? So when the protons concentration in the intermembrane space is higher than higher than the concentration of the protons inside the mitochondrial matrix, this is where the proton will be pumped back. The proton will pump back, flowing down or crossing this another protein so-called ATP synthase. Also, some say ATP synthetase, just the same enzyme. ATP, this is the final enzymes producer. Okay, enzyme producer, which embed in the electron transport chain, embed in the inner mitochondrial membrane. So the pumping of proton due to the higher concentration at the intermembrane space. So the proton from this space will be pumping back, cross this ATP synthase, this enzyme. It has a rotor. Nanti kita tengok, we'll look at later. Pumping back or flowing down back into the inner mitochondrial matrix. Okay, inner mitochondrial matrix. So the flowing or the pumping back of the protons through this ATP synthase, okay, it will stimulate or influence the combination or formation of ATP. Okay, it will stimulate the enzyme complex, because it's an enzyme, right? ATP synthase. It will stimulate the enzymes to make it complex with the substrate, which is inorganic phosphate. Okay, to combine with the ADP to form the ATP. Okay, due to the pumping of the protons through this ATP synthase. This is like a channel, a channel which spin the rotor, will spin the rotor to provide the energy, with for the enzymes to conjugate, to make their complex with the ADP, in between ADP and inorganic phosphate to form the ATP. This is where so-called chemiosmosis. Chemiosmosis, ATP synthesis powered by the flow of protons. So the pumping of proton flowing back down or flowing down here through the ATP synthase give the energy to form or to make the complex enzyme complex ADP and inorganic phosphate to synthesize the ATP. This is where all the electron which comes from the glycolysis, the Krebs cycle, being transported through the electron transport chain, being finally converted into the final ATP form. Alright, so this is called chemiosmosis. Due to the presence of the ATP synthase. Okay, the final enzyme producer. Okay, so the electron the same at the same kan at the same same location also on the ETC. So the electron is transformed from complex 1, 2, 3, and 4. So the final complex 4 is the final electron acceptor. Which convert the oxygen into the water. Okay, so it will stimulate the electron transfer, stimulate the protons that available from the mitochondrial matrix into the formation of water. Okay, so the complex number 4 is the electron acceptor. Electron acceptor to form the water molecules. So the electron transport chain, electron transport chain involve the electron transport as well as the pumping of proton. Not only the transfer the electron, but also pumping the protons, which creates, we all make the protons gradient across the membrane. That's why it will be pumping back into the mitochondrial matrix to provide to supply the energy for the ATP synthesis. Okay, to form the ATP. So both these process, oxidative process of electron transport chain, okay, together with the pumping of protons and the chemiosmosis, which involve the ATP synthesis, to produce the ATP or to convert the final ATP production, so-called oxidative phosphorylation. So the oxidative phosphorylation process actually transport all the electron from the Krebs cycle, okay, into the final ATP form. Okay, class, so it will involve the mitochondrial matrix, of course the ETC in the inner mitochondrial membrane, and intermembrane space. Okay, doesn't involve the outer membrane space, only the inner one lah, which close to the protein embed here. Okay, complex 1, 2, 3, and 4. All right. Okay, so this is the flow chart to summarize lah, the flow chart of the summarize of the electron transport chain. Complex 1, which is complex 1, FMN here, flavin mononucleotide, which receive the electron from an ADH, where the complex number 2 here, CoQ, receive the electron from FADH2. Right, so the electron from the FMN, from the complex 1, together with the electron from FADH will be further transformed into the complex 3, okay, complex 3 cytochrome B and C. Okay, sites of proton pumping coupled to ATP production lah. So complex 1, 3, and 4 only involve in pumping protons, except for complex number 2, CoQ. Okay, right. So complex cytochrome B and C transfer the electron until the final electron acceptor at the complex number 4. Okay, this is only the flow chart lah, which convert into the formation, which transform the formation of the oxygen. So the oxygen will combine with the protons in the mitochondrial matrix to form the water. Okay, all right. Let's look closer to this ATP synthesis or ATP synthesis class, yang the one that I show, this one, okay, this ATP synthesis or synthesis. Okay, so ATP synthesis or ATP synthesis make ATP from ADP and API. So how does ATP synthesis can make or can transform the ATP production? Okay, so they use proton gradient to power ATP. So this ATP synthesis, these enzymes need the power supply. So the power supplies comes from the gradient, the proton gradient from the intermembrane space. And because the electron transport chain involve pumping the protons from the inner mitochondrial matrix into the intermembrane space, which create the higher gradient of protons. Okay, so the higher concentration of proton in the intermembranous space will pump the protons back into the mitochondrial matrix through this channel of ATP synthesis. So the power source, so the protons tadi, the high gradient of the protons actually provide the power supply for the ATP synthesis when they are flowing down here through this channel. So the power source is the difference proton pump concentration between opposite side of inner membrane. So the process of chemiosmosis lah. So the chemiosmosis is the process of pumping back the protons from the higher gradient of proton into this through this ATP synthesis. And this is where defined by the chemiosmosis. So the energy stored in the form of protons gradient across a membrane is used to drive a cellular work lah. Okay, so the energy, the power energy from the proton gradient supply supply the energy to the ATP synthesis. So ATP synthesis or ATP synthesis is an enzyme protein complex which embed also in the intermembrane space. It has a rotor in the membrane spins clockwise. Ikut arah jam. Spin clockwise when how they can spin clockwise when the high gradient of protons flows through down, flows through it down. Okay, when the proton is pumping down, flowing down here through this, so the rotor on top here, on top structure here, it has a rotor. The structure so-called rotor will spin, spin. Okay, when the proton is pumping down. Okay, so a rod connecting. So there is a rod here in the center of the ATP synthesis. It has a rod which connecting the cylindrical rotor and the knob. Also spin also. Okay, the knob in between this rotor activate the catalytic sites in the knob. There is a rod that connect the rotor on top here and the catalytic site. Okay, so the knob which spin also, okay, activate the catalytic sites here, right? So what happens? So the protruding knob, the knob contain catalytic sites that will join the inorganic phosphate, as I mentioned just now. Organic phosphate, because it has these catalytic sites provide catalytic sites that receive the energy from the pumping of the protons, which stimulates, will make the rod in the center part spins, and spin and activate the catalytic sites. Okay, catalytic sites of the ATP synthesis ni lah to join the inorganic phosphate with ADP. Okay, to complete the transformation of the ATP. So just imagine, as long as just imagine lah, every second, this kind of oxidative phosphorylation, as long as we have alive, as long as we have alive, we have enough oxygen, whatever foods that we consume, okay, all the chemical energy being transformed into ATP. So every second of our life is actually running all these biochemical reactions until we get enough ATP. Okay, at the end of the process, and keep repeating as long as we're still alive lah. Alright, okay, so okay, class, I think because it's too much so far lah, too much. Okay, for this I think I plan to have to stop until here. Alright, so I want you to have a clear picture, overview of this process. From the fat, we are looking at fat later on. Later, okay, where all the small molecule fat, fatty acid lah, not fatty acid, become fatty acid now. So fatty acids from the body cell tu kan, fatty acids, glucose as well as amino acid lah. So the overview, how they are the same, they are all undergo the same, on just except for the first part. The breakdown of the fatty acid we'll see on the beta oxidation lah. But for the glycolysis, we have touched this, okay, and the product also the different lah, different with the beta oxidation, also different with the amination. Okay, but the flow, meaning that all the products, all the electrons from the glycolysis, from the beta oxidation, all being gathered and transport into the Krebs cycle. So we're producing the Krebs cycle is the same products. Okay, the same products. Okay, then converted fast to the electron, just the same process, okay, into ATP. So the calculation of the total numbers of ATP or will be depending on the numbers of NADH, FADH produced by the glycolysis, which is separately different with the beta oxidation, right? But the Krebs cycle is just the same, and so we can sum up lah at the end. All right. So the flow from the Krebs cycle, electron transportation or oxidative phosphorylation, just the same. Okay. So you need to remember the electron transportation, what are the complex, the sequence of the complex 1, 2, 3, 4. What are the purpose of electron and protons in ETC? Okay, and what is the function of the ATP synthesis in completing the ATP production, okay, through the chemiosmosis. Just the same. So class, we stop until here. The next 1 hour, you didn't decide yet kan? Belum kan, Aini and the rest? Belum decide 1 hour bila boleh buat. Nanti boleh you all discuss among you all. Nak replace yang 1 hour yesterday tu kan? Boleh ya, Aini and the rest? Class, you are still with me?
Yes, doctor.
Okay. Make sure you discuss among you all when the date and the time will be available lah for our class. I propose on Thursday. I propose on this Thursday afternoon. If you are free lah, tak apa. I let you discuss among you all. Okay, the next 1 hour though, we just look at only, okay, how the fatty acid, how the glucose, right? So how the fatty acid being broken down, through beta oxidation. Okay, only the beta oxidation. So, the overview from the Krebs cycle is just the same, but we just want to look at the very specific on how fatty acid, because remember fatty acid, it has, depending how long the chain length of fatty acid available, right? So it will involve different cycle, like glycolysis, it's only one pathway involve the 10 enzymatic reaction, right? Which convert into pyruvate acid. But the fatty acid, the fatty acid will undergo beta oxidation, which is a cycle. How many cycle depending how long the chain length of the fatty acid. So it will involve a few cycle lah, depending how long the chain length of fatty acid. Alright, so I want to discuss specific on this beta oxidation. It has a little bit different lah, because this is the cycle. Right, so we are focusing on this Thursday, inshallah, Thursday. And we calculate how many the total numbers of ATP being produced from different fatty acids. Okay, from different fatty acids. And then we discuss in our class, and I give you the tutorial lah. Just follow the same way. The same way. It's very important. I will be covered also in the final exam, right? So you must follow on this Thursday. Okay, so I give you example of the fatty acid with the specific chain length, and then just use the same. So I give you the formula lah, to design, how to calculate. Then I give you the tutorials lah. Okay, but if you can, you can try lah, before I teach you on this Thursday. Don't worry, But on Tuesday, make sure every one of you attend, our class, our final class lah kan. Alright. So, of course lah, beta oxidation, meaning that we still continue because this is the first part, beta oxidation, the breakdown of fatty acid, and then when it convert into acetyl CoA, then the same Krebs cycle will be will be involved lah, just the same as the glycolysis, right? So make sure after our class to our class, you make your own revision and the overview, starting from the molecule, small molecule, the stage 2 and stage 3 lah tadi, how the electron being transform, or the overview tu lah, the process tadi. Alright. Okay, so on this Thursday we just look at on the beta oxidation. How can it convert into acetyl CoA and then enter the Krebs cycle? Then you can see the differences, differences between the glycolysis and the beta oxidation. Okay, just the same, but the same on the Krebs cycle and electron transport chain, just the same. Okay, the difference of the products only from the glycolysis and the beta oxidation. Faham tak, class? Okay, so when we calculate, it's okay lah. Just get how many molecules of NADH, how many molecules of FADH, or GTP, and then we just time with the equivalent, with the times 2 ATP or times 3 ATP, or GTP times 1 ATP, and that's all. From the different, never mind, I will make it clear on this Tuesday to show the final, the beta oxidation, because beta oxidation, it has a complicated, a bit complicated lah, because it involves a few cycles, unlike the glycolysis, only one pathway and just the same, which is 12 ATP per cycle tadi kan, from the Krebs cycle, okay, ni sama, but the difference is part of this lah, the glycolysis berbeza sikit lah, the glycolysis produce 2 pyruvic acid, 2 and ADP, 2 ATP and 2 NADH only, no FADH, but for beta oxidation, I will show you, it depends how many cycles will produce different numbers, of NADH, and FADH. Okay, boleh kelas so far? Any question? Dah pukul berapa dah ni? Alright, okay. Okay, any question? Maybe you want to look back into a few previous slide ke? Ada tak? Or you want to kumpulkan until on this Thursday? Still sempat enough, only 1 hour to just focus on the beta oxidation and calculation. Sikit je calculation tu. Because we have done on the Krebs cycle, right? We have done. So, we look at, kita akan tengok, we will look back on this part lah nanti. So glycolysis have done this, we have only 2 NADH only, so you just minus 3 ATP, go 1 NADH times 3, equivalent to 3. Alright, but for beta oxidation will be different, but the rest just the same. The Krebs cycle, the numbers of products, just the same Krebs cycle. Okay, okay. So any question? Or I just share. Okay, any question class so far? Okay, tak sampai lagi 2 hours. We can ask. Okay, class. Apa? Okay, I open dulu. Any question class so far? Okay, very important for as biochemists, you know what are the location type, for different process, different pathway, biochemical pathways, from the glycolysis tadi, for beta oxidation a little bit different, so I don't want you to confuse today. So beta oxidation a bit different in terms of the location of the beta oxidation, it's a little bit different. So please get clear on this class today, the glucose, the glycolysis, the glucose breakdown too. So, and then how they can, the process lah how they can convert into acetyl-CoA to carbon from 3 pyruvate, become 2 carbon, acetyl-CoA, and then enter the Krebs cycle, involve the enzymatic reaction, right? And then what are the products, 3, 1, 1, 1, kita ingat, 2, CO2, okay, and then what are the shuttle go into, or transfer into the ATC, complex 1, 2, 3, and 4, and final converted through ATP synthesis. Okay. You can make your own story. Okay. Remember, the glycolysis involves 10 enzymatic reactions. There are 2 phases. The first input dengan output. So, but the net ATP is 2 ATP. But for the Krebs cycle, only involve 8 enzymatic reaction. Okay. Because even in the objective pun soalan objektif to find out the multiple choice question, you need to remember, I want to ask the enzymes, you need to remember how many enzymes, for example, involved, what are the products produced from glycolysis, contained from beta oxidation, depending on different types of chain length, and Krebs cycle, how many products. Okay, the numbering is very important. And then the different location type. Okay, itulah because this is the energy metabolism part lah for the digestion absorption. So you must remember many types of enzymes. It's a must lah. It's a must. Different types of enzymes. Of course, different location sites. What are the major resources? Where are the final location of the complete digestion and absorption of the nutrients? And the function of the accessory glands, right? Those things lah. And about the fat lipids kan very important because it's quite a bit complex sikit lah the apa tu, the lipids breakdown the lipid degradation kan or breakdown lah digestion lah, lipid digestion process in the small intestine kan, specifically in the the duodenum, remember duodenum. Okay, those things like whatever I mentioned in class will be asked in the final exam. Okay, secara overall lah. Okay, okay, ada 10 minutes more. Okay, now I open dulu. I open before we stop. Okay, one more. Sorry, before I terlupa. Okay. Previously I have mentioned I want to include the video punya ni kan. I want to include a few questions regarding on the video video presentation, right? The 5 minutes poster presentation. Okay, I want to make correction lah want to make correction. So the video we doesn't include in the final exam lah but only for the second paper tu adalah sikit second paper the set B. If you are not attending our final exam on 12 July kan, 12 July, so hopefully I pray all of you are healthy condition on the 12th July, so that all will be attending class. Otherwise class, we need to have the set B. So the set B question, set A doesn't include lah the video presentation, so you have done that. But for the set B, I include lah a few so but focus on the set A lah on the set A. Okay. Okay, you got me so I don't include the 5 minutes poster presentation in your final exam set A Okay, you got me? Class?
Yes, doctor.
Make sure you follow me You still with me kan? Rugi tau, rugi. Kalau you just ada depan ni, I tak tengok lah. I cannot see your face even though we are facing in front, but in front of the laptop. Okay, but this is a very important part. Energy metabolism, digestion, absorption are linked together. Digestion, absorption, and energy metabolism are very closely each other. Linkage, close linkage. Alright, so I hope you can get a clear topics for this topic. Okay, so I open to the question if any. Otherwise, we just continue on this Thursday and make tutorial. We can just try your own if you want on the tutorial. Kan? So never mind, I want to teach you first. But for those, if you want to try, you can do it first lah. Okay. Okay, 5 minutes more. Ada tak any question?
Doktor, kelas hari Khamis ni physical ke online?
I nak physical. Hopefully kan? Okay kan? Physical. Actually kan hari ni pun I nak physical but tak boleh lah. Okay, on Thursday, can we make it physical? Boleh?
Boleh.
Very important. Last class tau. Last class ni. Okay, tu ada tips tu lah. Senang, I rasa lebih prefer to see all of you face to face. And then I share also the what we call it, the format, the format for final exam. We have section A, B and C. Sama je, senang je Okay, so make sure you attend on this Thursday. Okay, hari Khamis tu okay, so fine. Nanti I ni ke boleh inform me the final decision lah. We try to make face to face. Inform me maybe hari ni ke, can you decide today so that by tomorrow ke, later by tomorrow ke, because I want to book, because this is out of our schedule class kan, venue, so I need to book first the any room, lecture room available on Thursday. So just let me know the timing on this Thursday, the timing only 1 hour. Okay kelas
Doktor.
Okay.
Doktor. Attendance hari ni macam mana ya?
Semua datang. So we have 38 now. Okay, nanti I minta you sign on the final. Itulah, the last day nanti you sign sajalah You sign, okay? Remind me. Boleh?
Boleh.
Okay. Last, any question? You hadamkan dulu Please digest first this topic energy metabolism and digestion absorption Okay. No more question? Please ask tanyalah I tak nak nanti bila I tengok kan the way you answer tu I macam tak faham. Looks like you don't understand. If you not remember, okay lah, I can accept. But not understand tu If you not understand, why you didn't ask me I have tried my best. Okay class, if there is no question, okay, please make your assessment, teaching assessment, remember. So we have been informed by faculty and university to remind the students. Ini also you can remind. I'm reminding today I'm reminding all of you to key in to complete your teaching assessment. Boleh kelas? Teaching assessment for me lah. Okay? Alright? Okay, for me to input lah. So, okay, that's all for our class today. If there is no any question.
Doktor, final masuk topik apa?
Ni lah, two topics, digestion absorption and this I mentioned tadi linkage kan in between digestion absorption and energy metabolism. These two topics only. Make sure you score, okay? Of course you can score. tak boleh, if you not understand, please ask on this Thursday. Of course, every time I pray for you, for all the students, I mentioned for my undergraduate student, my postgraduate, because only so far I have 8 master PhD students and final year project students. I pray for them, including you, insyaAllah. All right, see you on Thursday, and don't forget to make your teaching assessment today, as soon as possible today. And teaching assessment kan, boleh kan? You know, right? The link, and then let me know the date and the time on Tuesday class. Okay, class, see you on this Thursday. Thank you very much.
Thank you, Doctor.
Thank you, Doctor.
Okay.
Thank you, Doctor.